So here I'll try to cover uh, what we already know, which is what I call the old, what is happening currently in the field in terms of research, which is what I call as the new, and the future of the genetics research. So without much delay, let's jump directly into the topic. So, to begin with, principles of genetics, as we know so far from our textbooks, from our syllabus at the +2 and at the university level. So, I'll quickly introduce you to two books that help us understand the principle of genetics very well in our college system, which is Principles of Genetics by Robert Tamarin and Principles of Genetics by Snustad and Simons. Okay, and beyond this, we already have our syllabi teaching us the principle of genetics. If you follow this link, if there is an option to share this video later, that will be great. But otherwise, I can share my email ID to share this document with you all. So there is a lot of literature on the principles of genetics and where each of the terminology is defined, right? So if you follow this link, you will find all the necessary details. This is one very useful document that I refer to myself to go back to my basics. So once you start moving ahead in the field, you always start losing your ground. So you need to find your roots in terms of basics and strengthen your fundamentals. So I keep going to this document myself. It will also help you to understand all the basic terminologies related to genetics. So you can follow this there. But at the outset, a few things that I'll take from here for our session is what is a gene, right? A gene is a unit of inheritance that usually is directly responsible for a trait or character. So the genes, as we all have studied in our high school and our college system and our university system, this is what is responsible for our development, health, and also disease, right? They give us our characters. Either we are tall, either we are short, either we are healthy, or carry some genetic disease through our ancestors and through our parents. All that information is there in our gene, and we keep carrying it and passing it from one generation to the other. And alleles are two copies, one from each parent that we keep receiving. So they are copies of these genes. Alleles are nothing but copies of these genes. And homozygous means whether these alleles are same or different. So these details will come to in the lecture. I'll skip this document, but in essence, what we need to understand is we are what our genes make us, right? They give us the traits that we are tall, we are muscular, we have some metabolic disease. They define whether we have diabetes or we are not at all diabetic even after we age. All this is defined by our genes to a large extent. And to understand these principles of genetics, these are some of the recommended books to begin with. But let me touch on certain basics here, right? So we as humans have been evolving for millions of years, right? And with us, our genes have kept on changing, and our genes are present on the DNA, which is known as deoxyribonucleic acid, that is present on our chromosomes. And chromosomes has different parts like telomere, centromere, P-arm, Q-arm, chromatids. These are all basics. Let's not dwell too much time on basics since we all have been learning about this from our school curriculum, right? So this is the basic concept of the DNA, the deoxyribonucleic acid, which makes the chromosome. And here is the information that makes us all, that is present in terms of this sequence code. These sequence codes have genomic signature. These are signatures that helped us evolve into humans through millions of years of evolution from our ancestors, which are the monkeys, right? And as we all are reading a lot about engineering such genes, nature has already been doing it for us through evolution, either in a very slow pace in the case of humans or in a very fast pace in the case of bacteria. They also evolve. We all are prescribed antibiotics whenever we go to the doctor when we are sick, when we have some bacterial infection. We all get antibiotics prescribed to us, right? These are drugs that basically kill the bacteria. But over time, if you do a simple lab experiment, you will see that these bacteria over time evolve. They change their gene signatures to become resistant to antibiotic. They become immune to antibiotic. The same antibiotic that could kill them a week early cannot kill them anymore if you use the same antibiotic because they evolve rapidly in under a week's time in lab conditions or within our body too. This is the reason why we are suggested that we don't consume a lot of antibiotics unnecessarily because we will just make our bacteria that infect us more resistant. So you should not binge, not overdose on antibiotics, right? But the root cause of all this is evolution, where genes, their sequence or their arrangement, their rearrangement, all of this keeps changing through the course of evolution. Basically, everything points to changes in the genes, right? So we talk in terms of genes, which are present on the DNA that makes our chromosomes. Okay, this is the basic, as simple as it is, from humans to bacteria. And without their knowledge, our ancestors, through several thousands of years, even before this generation, they have been, in a way, scientists themselves, our ancestors thousands of years ago, who started selective breeding in agriculture and in animal husbandry, right? For instance, this particular plant species, Brassica oleracea, right? This is nothing but a mustard plant in the wild. This has been selectively bred for different traits, where now this same ancestral plant has been selectively bred for different traits, I said, right? For their buds, for the big size buds, which are now cabbages that we consume. They have been, uh, you know, selectively bred for lateral buds, which are Brussels sprouts that we consume in our diet right now. There is kohlrabi that comes for the thickness of its stem. There is leaf that grows in abundance in kale. In the case of broccoli, for stems and their flowers, it has been selectively bred. In the case of cauliflower, for flower clusters. All of these have their origins from this very wild mustard plant, which is present in the wild even now, right? So they have been selectively bred, so their genes are selectively passed to get these particular traits. Traits are nothing but characteristics like thick stem, long stem, uh, thick, long leaves, etc., etc., right? This is what happened with plant uh, selective breeding. In the case of animals, similarly, cows have been selectively bred for ages, where a good and heavy milk yielding cow has been selectively bred to get more milk yields generation after generation, right? That is how the whole dairy industry right now is relying on to get uh, high yields of milk to run the dairy industry, which is also a result of thousands of years of selective breeding of cows. Or when it comes to sheep or goat, it's for the meat. In the case of chicken, we get bigger varieties of chicken that give us more meat, and they eat less, but still make more meat in their body. Or in the case of our pets and domestic animals, like these dog breeds that have been selectively bred for aggressive or heavy built or cute looking furs, or small size and still cute looking. These are different, breeds of dogs that have been selectively bred over generation to get a particular characteristic which suits that owner. So this practice has been there for thousands of years, and our ancestors have been experts at this even before we advanced our science through industrial revolution in 19th, 20th, and 21st century, right? So this is quite an history to where we are right now. Let's understand where we are in the last century and the present century. So the field of genetics arose because of contributions from Mendel. Mendel was a priest and a scientist himself who discovered the dominant traits, dominant traits could be passed from one generation to the other by selectively breeding for these traits, and these dominant or recessive traits could be predicted using certain calculations based on what the parental traits were. So this opened the field for discussion that there is some inherent element in all of us that we pass on to the next generation, and these elements are conserved, where these elements mix in fertilization between these uh, germ lines, and then this helps in getting the right kind of progeny. And they didn't know what these elements are until the discovery of DNA and its structure, right? And then came the DNA double helical structure that fascinated everybody, where using certain crystallographic studies, Rosalind Franklin demonstrated through her data that DNA could be double helix, which is what Frankis and Crick used for their Nobel Prize winning recognition. But it's debated why wasn't Rosalind Franklin, who generated this data, was not awarded the same prize. And it's quite controversial even to this date. But moving on from here, we have one of our own, Hargobind Khurana, who contributed immensely to one of the many discoveries in genetics, along with Robert W. Hawley and Marsh W. Nirenberg, right? So the contribution these gentlemen made were through discovery of these genetic code as to what three-letter code could code for a particular amino acid. So our genes, as I said in the beginning, are made of these four letter code: G for guanine, C for cytosine, U for uracil, A for adenine, right? These U in RNA and T in DNA, right? So these codes, these triplet codes, three-letter codes, can be understood in this manner, where you have UUU, UUU coding for phenylalanine, and UCU coding for serine, and so on. If you follow this table, you can understand what each of these three-letter codes stand for. In the case of GCU, let's understand GCU, it's alanine. So GCU codes for alanine, and ACG, let's understand ACG, ACG codes for arginine, right? So in this manner, if you follow these three-letter codes in our transcripts, you will understand what is the protein sequence that will follow this coding sequence. Each of these will code for one amino acid in the protein coding sequence. And this information is is flown from DNA to RNA into proteins. We will understand these in one of the images downstream. If you have any doubts, it will become more clearer in the next couple of slides. Now, one of the many interesting discoveries in genetics was the discovery of polymerase chain reaction as well, where the idea that DNA replicates in the cell, right? So our DNA replicates in the cell using a replication mechanism where an enzyme known as polymerase, which is what is used in this polymerase chain reaction to artificially make DNA, is also what is used by our cells to naturally replicate, make new DNA copies from old DNA copies. Whenever the cells divide and form new cells, our DNA is also replicated to copy the code into new cells, right? So this understanding of the DNA code that is being replicated into new cells, where more DNA is made as copies, was used artificially in the lab in a technology known as polymerase chain reaction, where you could use DNA as a template, right? Just like your Microsoft Word template, using which you will build more interesting slides for your presentation, or just like your template application form from which you will fill in your biological data, right? So just that way, one of the copy of DNA is enough to make more copies of DNA like this. From one copy, you make hundreds or thousands or millions of copies like this using this technique known as polymerase chain reaction. In this, what happens is, in the first cycle, you use these primers which partially recognize the DNA sequence on the opposite strands, from which the polymerase and the nucleotides AGTC will extend these chains and make more copies of the same material. And this material made from one chain will be used to make the next copies. And these copies will be used to make more copies in this chain reaction. And this relies on this enzyme polymerase that will be used to make these copies. And this was one of the most important inventions in genetics and molecular biology, using which you could make artificially copies of DNA sequence or genes from a DNA. And this is what our DNA fingerprinting or forensic labs use to test our DNA. This is what our COVID testing also uses in a way, but not the DNA. They make DNA from RNA, which is also built on this PCR itself, right? Where there is this polymerase chain reaction um, that is happening. And this forms the basis of many experiments and discoveries in genetics and molecular biology. So in this way, discovery after discovery paved the way for understanding genetics better. One of the other discoveries that happened and led to a lot of significant development in genetics is the sequencing of the DNA itself. Here, the same polymerase that is used to amplify DNA in PCR is again used to um, to extend these chains of DNA, these strands of DNA, but you add a nucleotide in the reaction that stops the reaction abruptly. And when you size separate these nucleotides, where each of these nucleotides are labeled in different color codes, right? DD and TPs. right? So if we zoom in here, you can see that each of these DDNTPs, they have a D-deoxy, which will stop the reaction from growing, right? They have DD-TTP, DD-CTP. So each of these is again ATGC, adenine, guanine, thymine, cytosine, and TP. Um, so these DDTPs will participate in this primer elongation and chain termination. And when you size separate on a capillary, these will segregate themselves according to the size, which you can read on this platform where you have a laser that will detect what nucleotide. So these nucleotides are labeled with fluorophores, meaning that they emit light when you shine laser on them. And as they are running through this in a size gradient manner, as they are passing through this capillary gel in a size gradient manner, when you apply electric field, they are detected by shining laser, which will emit these wavelengths of lights, right? Different wavelengths of lights, depending on what they are labeled with. And each of these are also detected digitally using the detector that is connected to a computer, and these generate these peaks. These peaks are read in the form of a chromatogram, where each of these color codes that will be detected and passed to the computer, this information will be digitally read as your DNA sequence, which all originates in this experiment of primer extension in this PCR reaction, right? So this forms the basis of all the DNA sequencing. So this is also the basis of the DNA sequencing to detect mutants of coronaviruses that are doing rounds now. And these are what are used to detect mutants and variants such as Omicron, Delta, and its earlier variants too, right? So this is the modern Sanger sequencing. So the discoverer of this technology and its earlier version was Sanger. And in his technology, one had to read these sequences in a tedious manner using gels, and it used to take days and weeks to read the entire length of DNA. But with advancement of technology now, we can fluorophore tag, read them on a gel by detecting it and with help of computers. Before the development of computers, all you had to do was run it on a gel, applying electric field, and then detect what these nucleotides are using this methodology where primers are radio labeled, right? Or the DNTPs are radio labeled. And they would detect based on the size separation, just like on capillary gel now. But this was taking weeks and months to generate data. Now, this happens within an hour or two in the modern Sanger sequences. And even their advancements with next generation sequencing machines like NGS, as it is popularly referred to as, they can read millions. And actually, the depth is now in giga, giga terms. So it's not just million, right? So these are next generation sequencers and nanopore sequencing, which have advanced technology to read the sequences in clusters. So for the convenience of the audience, I'm not going into next generation sequencing. So in sequencing, what you read is basically the nucleic acid sequence in the DNA to understand what is the code and what has changed from one organism to other, from one person to another, from one virus to other or bacteria to other, right? This is the way you can genotype an organism. So this is called genotyping. to understand what is the gene sequence. And for it, you use DNA sequencing as the ultimate technology. This was one of the prominent discoveries in genetics. This helped us sequence the human genome. This helped us sequence a lot of pathogenic bacterial genomes and also mouse, rat, and zebrafish genomes, which all help us understand the basics of genetics and molecular biology by using them as models. We'll come to these details in a couple of slides. So this slide is very important to understand different omic layers that regulate development and disease, right? So all of these are beyond the fundamentals of the principles of genetics. So like I said, principles of genetics can be picked from those textbooks, and already basics is being taught in high school and college or university systems. So I'll not go into the basics much. I'm talking about the next level of things here. And the next level of things rest in these four layers. So the genome, where the information is at the level of DNA. The DNA make the RNA in a process known as transcription, where the DNA is transcribed into RNA. Deoxyribonucleic acid is transcribed into ribonucleic acid. And this RNA, this level of information where there is RNA is called transcriptome, because these are transcripts coming in from DNA. So they are called transcriptome, right? The level of information coming from DNA is called genome. And then there is translatome. This is an up and emerging.
One
Yes.
thing.
Yeah.
Dr. Gnaneshwar, can you change this actually this cursor, your pointer, na? Little bit not
Okay.
small size, something like that.
It's not happening with this file for some reason today.
Okay.
Sorry about that. Is it inconvenient?
It's okay. Okay. Somebody asked to actually size it that pointer. Can
Okay. Yeah. This option is not working today. Sorry about that.
please carry.
Okay. All right. So I'll try to minimize the issue with this pointer here. Okay, I'll keep it away from the data that I'm referring to. Okay, so I was talking about genome, transcriptome, and then translatome. So this is a very crucial level of information where what our DNA transcribed as RNA is now translating into proteins, right? The RNA has come in on ribosomes and they are translating as proteins, translating into proteins. So the RNA, which is the messenger RNA, now becomes proteins on ribosome. And this is a crucial regulatory mechanism that is being studied since last 1 decade. And there is a lot of control in biological systems at this level where it's not what is there on DNA. It's not what the DNA makes, but it is what it is at this level that translates into proteins that is very crucial for biological systems along with these different layers. Okay. Now, once the proteins are made here, these proteins are referred to as proteome, right? This is the final layer of information. This is one final layer of major information in biological systems, where proteins make most of the cellular components in an organism. So you basically have genome, transcriptome, translatome, proteome. These four levels that heavily make the biological systems. And these are very important to be dissected in this session. You will understand why, because we will talk about what influences the outcomes beyond principles of genetics. So principles of genetics is a very well laid out field that paved way for these advances. And now let's talk about the advances that have come out from following these principles of genetics, right? So the first advance that came out of these findings is epigenetics, where you study how behaviors and environment can cause changes that affect the way your genes work. And unlike genetic changes, where you talk about nucleic acid changes that dictate what protein is made or what is the protein sequence, you talk here in terms of what gene is on, what gene is off, and how these are regulated. You talk in terms of gene expression as they also refer to it, right? What gene turns on, what gene turns off. And an important regulatory mechanism that regulates this is non-coding RNA. So what makes these proteins are the coding RNA, which is the messenger RNA, right? They are the coding RNA that make these proteins. There are certain RNAs, and in fact, it has come to realization through last 2 decades of research that more than what we refer to as coding RNA, there is a lot of non-coding RNA being made by our genome. Okay, so these non-coding RNAs are very important in epigenetics. Some of these are small non-coding RNAs, lesser than 200 nucleotides. Okay, and the other are long non-coding RNAs, which are greater than 200 nucleotides in length. So there are non-coding RNAs which do not code for proteins, which regulate the gene and its turning on and off for expressing this flow of information in biological systems. So one regulation that is important for these epigenetics to function is methylation of DNA. So methyl group is added to DNA to block proteins. So your proteins require certain factors to bind to them to make RNA. Certain proteins have to bind to DNA to make RNA, and RNA have to further become proteins. And some of these proteins again come back to this cycle to attach to DNA and make more RNA, right? So that process of protein binding to DNA and making RNA is blocked by methylation. And this turns off the gene. And reversing this process is called as demethylation, which turns on the gene expression, which methylation blocks this process of flow of genetic information. Demethylation, removal of methyl from certain places, wherever those gene sequences are, turns on this flow, right? And there is histone modification, where your chromatin, where your chromosomes make a knot around these histones, which get acetylated, methylated, phosphorylated, deaminated, or ubiquitinated, right? So these are 4 processes where there are chemical modifications to histone. As such, histone DNA sequence wrapping around histone like these will block the genes in those sequences at those points from their expression. So your genes are turned off whenever histones are attached to those DNA sequences and DNA wraps around it. And there is, on top of it, different chemical modifications that further influence how these histones help or not help in making these genes turn on to make RNA, right? So this is another level of regulation in epigenetics besides DNA methylation. Now comes non-coding RNA, right? So for DNA methylation or histone modification, non-coding RNAs participate in these processes. So it is the non-coding RNA, the RNA that doesn't make any protein, that is what participates in these processes. And such RNA are four different kinds. These siRNA, microRNAs or miRNA, pi PV interacting RNAs, and link, long non-coding RNA. The first three are small non-coding RNA. The last one is long non-coding RNA, greater than 200 nucleotides of ATGCs in length. And they have several characteristics and they influence certain processes, right? Like silencing or transposon repression, where DNA is methylated, or genomic imprinting for X chromosome inactivation. And these RNAs come out from DNA as primary microRNA. For instance, this is the pathway that makes siRNA and microRNA, right? So microRNA genes transcribe these microRNAs. They are processed using DROSHA, exported to cytoplasm from nucleus. They come to cytoplasm. They are processed into duplexes of microRNA, which makes a single strand of microRNA. They have base complementarity to mRNA that go and translate into proteins. Now they stop these mRNA from making proteins, or they degrade, meaning destroy these mRNA from making proteins. So this non-coding RNA, these non-coding RNAs now are able to regulate genome expression, right? Either they methylate or histone modify at these DNA sites, or they destroy these mRNA or stop these mRNA from making proteins. So this is what they are capable of, these non-coding RNAs. And they regulate the genome and hence influence the outcome from your basic understanding of genetics, right? Whatever dominant recessive trait, when you don't observe as expected through your principles of genetics, it is because of these non-coding RNAs, all of which influence the genome, the way it expresses to make this flow of information into RNA and proteins. So this is the influence of epigenetics, and some people refer to this as our biochemical karma in biology. Okay, this forms the basis of transfer of information from one generation to other generation. For example, in this Dutch hunger winter famine, where people who were exposed to famine, especially mothers who were pregnant during the famine, they were seen to give rise to babies who have heart disease, schizophrenia, diabetes, and even after 60 years of that famine, they looked at these epigenetic regulatory mechanisms such as methylation or histone modifications, etc., etc. And in this case, they saw that such modifications were present even in babies, and those were passed on to generations which were susceptible to these diseases because their parents or their grandparents were exposed to famine and starvation, right? And that effect was passed on to future generation, as we referred to in mythologies. and spirituality as karma. But I'll give you a disclaimer here that this is a non-scientific analogy. I'm just using this as an analogy. This is not at all an example. My reference to here or even online or in literature, they refer to this as some sort of biochemical karma. This is not scientifically proven. Karma, for your kind attention, is not scientifically proven process. This is just an analogy uh, to make a better parallel storyline to make this point be understandable to the audience. Okay, so this sort of epigenetic regulation where whatever alleles are passed on from one generation to another, in contrast to what the alleles are passed, you have diseases. Although the alleles were healthy, there are still diseases in other generations, right? So the grandchildren were not exposed to famine themselves. It's their grandparents who were exposed to famine. And famines are not supposed to affect genes of people, but still the effect is trickling down through generations. And that is because of epigenetics in play, where the expression of either non-coding RNA or these different non-coding RNAs is influenced. And such an understanding is being currently sought through research, where they are trying to understand the epigenetic basis of many diseases, such as neurodegenerative disorders, cancers, diabetes, and many rare diseases. Or in normal development, aging, health, and metabolism, these epigenetic regulations are still in play. Now, this is, like I said and emphasized in the beginning, one of the most critical layers where the information from genes to proteins, we all understand that genes pave the way for formation of proteins, right? But this translatome and mRNA translation into protein is the bottleneck for this flow of information from genes to proteins, right? Because mRNA is one translated into proteins, and not all that is transcribed is translated into proteins on ribosome, right? Most of it will not be translated into protein. That is why when people tried to quantify by numbers what the genes make and how many proteins are to be available downstream using omic method, they see a mismatch between what is being transcribed as RNA and what is being converted as protein. The bottleneck for that and the reason for that is here, where mRNA that becomes protein is controlled on ribosome. Let's understand this um, deviation from the principles of genetics, right? So ribosomes, these are ribosomes where two units of these ribosomes, the large and the small, come together. Between these ribosomes, the mRNA gets stuck and mRNA is translated into protein, where the genetic code is read and corresponding amino acid is added, where ATGC is read into corresponding amino acid, right? Like alanine, phenylalanine, serine, etc., etc. And these proteins grow in length from ribosomes to make a complete protein sequence. And this is a very important process, as you will understand. This is where the genes become proteins. And this ribosome is made of 80 different ribosomal proteins and 4 ribosomal RNA. It's made by a variety of things, and it is assembled by 200 factors, and mRNA is translated into proteins on these ribosomes with help from 200 additional factors. So there is a lot of events that control what mRNA becomes what protein. Literally, half a thousand factors can regulate what becomes a protein at this step. And so don't be overwhelmed with this diagram. This is to just show how complex that process of protein translation is. And most of it, or at least some of it, you all will be familiar with, where these translation initiation factors, elongation factors, termination factors are all coming on ribosome to translate in biochemical reactions to translate mRNA into proteins, right? So the idea of the slide is to drive the point that a variety of factors play a role in making proteins on ribosomes from simple mRNA that came from our DNA. Now, once we understand that there is this variety of information that becomes proteins, it becomes it becomes very important to understand what is translating or what is not translating, because that clearly becomes the protein. So people devised ways, devised methodology and techniques to understand this, what mRNA becomes what protein, right? By capturing ribosomes and looking at the mRNA that are stuck on ribosomes and what translates into these proteins on ribosomes. So to do that, um, so in our lab, we use zebrafish as an alternate preclinical model to study human conditions or whatever human biology we need to understand. We use zebrafish as our platform to understand that. And in the field, it is used as a model animal to understand heart, brain, vision, digestion, kidney systems, blood circulation, even development, and many other um, uh, phenomena, right? Many other events that is relevant to biology. This is because 70% of protein coding human genes are related to zebrafish genes. So there is 70% identity between human and zebrafish genes. And those genes that are important for human diseases, 84% of them have a zebrafish counterpart. So those genes that are disease causing in humans also have a copy of such genes in zebrafish. And zebrafish lay a large number of eggs, which helps in scaling up your study. And the development is very quick. By 5 days, they have um, eye, heart, brain, kidneys, pancreas, liver, all of it developing by 5 days, embryonic stages. So this helps in using it as a quick as an animal model that gives you quick data to understand biology and genetics. So in our lab, we use zebrafish where we have tagged the ribosomes that I was talking about. So these ribosomes that translate mRNA into proteins are tagged in specific cells of zebrafish known as Purkinje neurons in their brain. So wherever this ribosome is tagged and expressed, it it helps reflect a light of certain wavelength. And I have imaged that. And here we have used a vector and transgenic system to express this, where we can drive the tissue specific element that is only expressed in these cells. We use it to express this tag of green fluorescent protein that is shown here fused to ribosomes, right? RPL10 is a part of ribosome, and this fusion is expressed only in these cells in the brain of zebrafish. And these cells help validate that ribosomes are tagged in these cells, and I could image them. What we did is, so if this is the zebrafish larvae, and those green regions are the cells in the brain of zebrafish larvae, we have made a soup of these larvae to pull those ribosomes from only those tissues. And we have made a library of you know, translating mRNA stuck on ribosomes. Because we pull the ribosome down using our proprietary method, we are able to capture what is translating into proteins, right? That's the beauty of this technique. We are using it for sequencing these mRNA that are translating into protein, not just any free-floating mRNA. These are what becomes the protein. And we are using it for uh, next generation sequencing that I explained to you in the beginning of this slide, to do bioinformatic analysis, to basically tell what are the mRNA that are translating and what are not translating into proteins from these tissues. This will help us understand many different neuronal functions in these mutants that we have made in the lab, one of which I will tell you in a while. So in these mutants, by using these mutants, gap junction delta 2B, autism related mutant, or dopamine related mutant, or chemical treatment, what we will get to know is in these specific population of cells, what is becoming protein or not becoming protein in that disease system, right? It is very important to understand if you have a disease system, what is the profile of biological molecules such as mRNA, such as mRNA translating into proteins or the proteins themselves. What is becoming a protein or not becoming a protein in health and disease? That profile is very important for understanding disease and also to come up with treatment. So we are currently doing this project to sequence and tell what is happening in these animal cells, and only those cells, and not any other cell, but only those cells, right? So I'll not, so this is to just show that the technique is working, and we improvised it on a previous technique, and this is the control where the pull down is lacking, right? So this is the ribosome that is being pulled down, where the ribosome is intact compared to a previous method where ribosomes are degrading, meaning destroyed. In a previous method, we have improvised this method because the ribosome is intact and not broken into fragments. That is what this data tells. So this is called the bioanalyzer profile. This is called the bioanalyzer profile of ribosomal RNA that is stuck, that makes the ribosome. So this bioanalyzer profile is telling us that. Next, we used our sample to do a very sensitive PCR, which is one of the most advanced PCRs now, which is called Droplet Digital Pcr. This is an improvement from RT-PCR that we all keep hearing about, right? So this is Droplet Digital PCR, where we show that using our pull down method, we could capture some transcripts that were translating, which we could show here, and not in the control. There were 0 in the control. So this effect tells us that our technology is working, and we can use our technology to quantify what is translating into proteins or not translating into proteins, where you can use it to understand what is happening in diseases such as neurodegenerative, cancer, diabetes, rare diseases, or even in normal development and health, right? So there are different techniques, different such ribosome profiling techniques, where you can capture the ribosome that is translating mRNA into proteins and then tell what is happening in these cells, just like our technique that we spoke about. All of these come under translatomics, right? So this is an emerging field, like I told you. It's been studied from since a decade, and they have seen that a lot of changes happen at translatome, which is here, where ribosomes make proteins from mRNA. And this is being used in all of these disease and developmental states to tell what is translating or not translating. Okay. And the next level of information is at the proteome, where whatever has become protein is stored and is used by cells to build the cell. This is the proteome, which can be profiled using mass spectrometry and peptide sequencing. I'll not go into this in the interest of time here, because whatever we have studied at these levels, the genes, the transcripts, the translatome, all of it making proteins will again influence the genes, right, in epigenetics, or what becomes the protein. And this flow of information and its regulation at various levels that I just told you is very important. And these are not what the principle of genetics cover. And the reason I am doing this session, where it is important to make you understand that there are things happening in the field beyond the principles of genetics that is being taught to all of us, will help us advance our knowledge, bring us up to speed with what is happening currently in the field. And so we understood that all of these levels of changes are happening in disease and development, and we are trying to study that, right? So to understand this better, it is important to engineer these genes to understand uh, events that are happening beyond the principles of genetics, right? So engineering the genes using appropriate tools is very important. And to do this, it is important to understand certain examples, such as coronavirus, which keeps, as I spoke to you in the beginning, it keeps evolving, meaning that it keeps changing its sequence information to make it better recognize human cell receptors, which help them recognize human cell receptors so that they can gain access into human cells. That is what the coronavirus is doing, and that is what most of these mutants and variants of coronaviruses are doing to better infect us. So this has been happening in nature, and such genes of viruses have been invading human genome and inserting themselves in the human genome, and 8% of our genes are already made of virus genes, right? So this we know, and even coronavirus does the same, where it integrates itself in our genome. Nobody knows its effect, but this is the latest report that says that coronavirus, yes, they can insert it themselves in our genome, right? The pink is the coronavirus genome and the green is human genome. And through sequencing, they found that it has integrated into human genome. Now, if you were to reverse these effects, any of these effects, or in any of the diseases that are caused by genetic defects, right? You need to use certain tools. One of the tools which was awarded the Nobel Prize in Chemistry in 2020 was the CRISPR-Cas, where these two scientists, Emmanuelle Charpentier and Jennifer Doudna, received this Nobel Prize for the discovery of CRISPR-Cas. Let's just quickly understand what this CRISPR-Cas is, right? So CRISPR-Cas is basically a bacterial defense mechanism, immune mechanism to protect itself from viruses. The bacteria have CRISPR loci and Cas genes that copy the viral DNA and store in bacterial CRISPR loci to make CRISPR RNA that will again bind to these same enzymes and go attack the same viral DNA. Basically, virus infects us through their DNA or RNA. These CRISPR Cases go and attack those virus DNA or RNA because that information was stored by bacteria, right? And this cycle of events happen whenever the same virus tries to infect the bacteria. And this discovery of clustered, regularly interspaced, short palindromic repeats that stand for CRISPR was awarded Nobel Prize in Chemistry in 2020. And this has become a tool to edit any gene. So here the discovery was in bacteria. So people just take this and apply it to mammalian cells or plant cells or even bacterial cells to just edit any piece of DNA by sending the target CRISPRs to the target. These guide molecules are the CRISPRs that go to the target and remove the target strand and replace it with a healthy strand. Let's just quickly understand why it is required in genetics or molecular biological work, right? There are always these mutations that keep happening in our cells, transitions or transversions where pyrimidine is converted to pyrimidine or pyrimidine to a purine. And these result in silent mutation where the amino acid code is not affected. But in nonsense mutations, they affect the amino acid being added to the protein because stop codon comes in, or missense mutation where one amino acid changes into another amino acid because a nucleotide is changed, or frame shift where the frame is changed, where this sequence is completely different from normal sequence, right? All of these are the basis of majority of human genetic diseases, and you want to reverse them using a tool like CRISPR-Cas. That is why CRISPR-Cas is so very famous. And also, it is important to use these tools in reverse genetic approach, where you want to understand a given gene's function in giving a phenotype. So phenotype is the trait, the characteristic of cell or an organism that arises from the genotype. A certain sequence of gene can give rise to a certain characteristic, shape, morphology, etc., of an organism that is referred to as phenotype, right? So you want to understand what gene or what sequence causes this normal or disease effects, and you want to understand it using reverse genetics, where you tweak the gene, where you modify the gene using CRISPR-Cas as one of the tools to understand how it is important in maintaining a phenotype, right? And there is forward genetics also. You have the phenotype and you want to understand what are the genes. So that is forward genetics approach. I'll not touch on this to not induce confusions here. Let's just look at CRISPR-Cas because this is currently hot and happening area. And to understand CRISPR-Cas, here what CRISPR-Cas does is these guide RNAs that have target specific CRISPR RNA sequence. It recognizes a certain DNA sequence as a target through base complementarity. Adenine matches with thymine, guanine with cytosine. This is the basis of genetics that we have understood from our college high school system, right? It's the same mechanism with which CRISPR-Cas, if there is a here, it recognizes T. Adenine binds to thymine on DNA and so on, guanine to cytosine. And these four-letter codes keep repeating in keeping with this DNA sequence for complementarity, right? So this base complementarity is maintained in the guide RNA to target to a specific portion of DNA where it brings the Cas9 enzyme. It's a endonuclease enzyme which cuts the DNA like this, right? When the guide RNA with this stem loop structure comes with this Cas9 enzyme to the DNA, it cuts the DNA. When the DNA is cut, it opens it for mutations or specific precise editing, whatever you want to do. And this is the basis of CRISPR-Cas9 and the simplicity of its structure and function helps a lot of people use it and apply it in genetics to reverse the genetics and then understand whether it is good or bad, whether you can use it in treatment or its role in disease. So I'll address this only if it is required. So this is the three-dimensional structure of the Cas enzyme. And there is this green color RNA embedded in its structure. And this helps it activate and go bind to the target DNA where it where the RNA to RNA binding happens, and Cas9 recognizes certain three-letter code in the DNA. And both of them now cut the DNA into pieces. And it is a very specific reaction that happens here. If there is no match, it doesn't cut random sequences of DNA. Okay. This is used in what people refer to as genome engineering, gene editing, and recombinant technology to reverse the effect in genetics. And this is what we also used to generate mutants using its previous generation tool referred to as TALEN. So what TALENs do is, just like guide RNA, these are proteins that recognize specific sequence of DNA. And there is FOC1, just like Cas9 and CRISPR-Cas9 system. We used this system referred to as TALEN, which is a DNA binding protein-based system. It's a DNA binding protein, TALEN, which recognizes specific sequences of DNA on opposite strands like this, and FOC1 enzyme cuts the DNA. So this is what we used in our study, like this, TALEN1, TALEN2, FOC1 enzyme, to target a particular neuronal gene referred to as gap junction delta 2B to make mutants with this mutation that adds G, CTG, CGA, right? So CTC-GAG is converted into CTGC-GAG. Now the site is mutated in this gene that shifts the protein coding sequence and stops it abruptly here in our mutants, as shown in the last two lines. And these mutants are genotyped using DNA sequencing here that I described to you earlier, and also immunohistochemistry, where you check for this protein presence or absence in mutants, right, and quantify them, right? So this is what we did in our mutants, where we show by PCR, and also at the DNA level, there is mutation, and also at the protein level, the protein is lost in these zebrafish. And we use this in our study to understand during development what happens to these neurons, where neurons connect with each other to form electrical synapses, where electric uh, signals are passed from one neuron to another. And this we understood, if you make these mutants, their electrical and chemical signaling between these neurons are affected, because of which the development is affected in these neurons in our study, which we published recently in eLife. So one of the main application of this CRISPR-Cas is in beating the Mendelian inheritance, right? So we all study about Mendelian inheritance in principles of genetics, where in normal inheritance, you have a certain probability of your copy. Like for instance, if the red color of this fly is to be seen in the next generation, there is a 50-50 chance, right? 50-50 chance in normal Mendelian inheritance for these flies to be either blue or red. There is 50-50 chance. But using CRISPR-Cas based gene drives, you can make 100% probability of infiltration of this trait, the red color gene in these flies can become 100% in these populations over generation. And you can silence the wild type gene completely or knock it out from these flies. If you apply the same to, let's say, mosquitoes that carry these malarial, Zika, or other parasites or viruses, you can remove them from our environment, which causes a lot of damage to human population. I'll come to the consequences later, but what happens in CRISPR-Cas is, so CRISPR guide RNA and Cas9 is generated in one of the allele. We all have two alleles, right? One from the mother, the other from the father, right? So one of the alleles will have CRISPR-Cas9, and you let out such mutants in the wild. And whenever they mate with their wild type counterparts, what happens is this CRISPR-Cas9 cuts its complement, its counterpart, another allele of the same gene. It cuts the allele and it inserts itself. So this mutant will cut the opposite, the counterpart allele and integrate itself. And this makes 100% penetration possible in a population for a particular trait, right? This is a very powerful technique that is used to make 100% penetration in a population, and its use is being debated for its application in removing certain pests that are problematic, that impact environment, economy, and society, right? Basically, you are right now relying on shooting, trapping, or chemicals, or biological threats. But here, using gene drive, you can silently eliminate a population or revive an endangered species. One is to remove a species or to revive an endangered species. So this is the same mechanism that I just spoke of, where these gametes are fertilizing, and in that process, these CRISPR-Cas allele from one chromosome is used by the other chromosome by this cut and stitch mechanism, which could drive the population to either extinction or revival in 100% manner, right? So this is being debated to remove mosquitoes that carry viruses or pests that affect the farms or nature, to remove weeds from the nature, or to revive endangered species. But here, the issue is that you will be playing the god, right? And what if the control is lost somewhere down in evolution, and the species populate themselves more than expected if you are trying to revive an endangered species, or the loss of a particular species affects the environment in terms of its food chain, or other consequences, such as climate change, right? Climate also depends a lot on particular species in that environment. So we don't have a handle on such complicated hierarchy in environment and ecosystems. So this technique is right now put on halt across the world. It was also thought of being implemented in India to eradicate these mosquito-borne diseases, but it's, in my understanding, put on hold. So the other important application of this CRISPR-Cas is in biomedicine, right? You can treat a lot of diseases where DNA mutation is in play, such as cardiovascular diseases, eye diseases, lung, brain, heart, etc., etc. And there is a lot of preclinical trial in animals that is already happening for thalassemia, hemoglobinopathies, congenital amaurosis, Duchenne muscular dystrophy, and so on. Endless list. It is growing for its application to edit genes in animals, and some of it has already moved to humans as well, where clinical trials are ongoing, such as beta thalassemia, sickle cell disease, and Leber congenital amaurosis, where patients have been recruited by pharmaceutical companies to edit their genes so that they can cure a certain disease, right? And diseases such as cancers are also to benefit from CRISPR-Cas, where multiple genes are affected in cancers, right? Cancer is not just one gene, one disease. Many genes are affected in cancer. With CRISPR-Cas, the simplicity and its ability to target multiple genes in one go, you have a good shot at curing cancer as well. And all these are being tried in human patients where cells are taken outside the body, genes are edited using CRISPR-Cas, and these cells with genes edited are being put back in patients, in these cancer patients, such as these peripheral blood lymphocytes, where T cells are silenced for a particular cancer-causing gene. And controversially, I'll be done in a while. Controversially, CRISPR-Cas babies were also generated without approval in China by the scientist He Jiankui. He generated babies by using CRISPR-Cas by editing certain AIDS, HIV/AIDS related gene, and those babies are they are healthy and doing fine. That's the report, but it's kept very confidential. And he was penalized for editing human genes in germline systems and making babies out of it. For this unethical behavior, he's being penalized by the Chinese government. And many countries have signed a legislation to not edit human embryos for research. It is allowed with approval, but not for practice to make human babies as such, right? So this is the current status of CRISPR-Cas. There is a lot of potential to treat diseases and balance health using this CRISPR-Cas. But the question is, will it be used for human enhancement, make better humans? That's for the future to decide. But in summary, what I discuss with you here is that genetics is a very broad area with subspecializations in themselves, such as epigenetics, which influence gene expression, transcriptome events that are also involved in regulating epigenetics and gene expression, translation, which is a crux in flow of genes, genetic information into proteins, and then proteome that is finally readjusted for stability and expression, and that we can genome edit using CRISPR-Cas, which can change the course of genetics related applications in health and disease, right? So with this, I thank you all for your patience.